One important fuel for the cell is glucose. The body acquires glucose by breaking down carbohydrates in the diet into their simplest form, which is often glucose. Glucose is converted into this molecule, pyruvate, through a series of steps known as glycolysis. We'll look at those steps in a later tutorial. The process of glycolysis produces two of these ATP molecules. It also produces two NADH molecules. These NADH molecules will be converted into ATP later. The pyruvate molecule then undergoes another series of reactions known as the tricarboxylic acid cycle.
We have an acetyl group. We have a 2-carbon molecule. When we rip this carbon dioxide off, energy is created, energy is released, and we use that energy to reduce NAD, reduce this coenzyme, to make NADH. And we know NADH then carries energy to the electron transport chain, because that's the big dog in this situation. That it's going to make the most of our ATP. So now we have an acetyl group. We're continuing to rip hydrogens off of it. We're continuing to destabilize it. And speaking of destabilization, a coenzyme A is also going to be added to it. And coenzymes, when you add them to molecules, they make them extremely unstable. So we have this acetyl group, these red carbons, which were originally from the glucose. We're continuing to metabolize that original glucose. We're down to 2 carbons, but recall this happens twice. Okay, 2 pyruvates from 1 glucose, so this is going to happen twice. So we have 2 of these acetyl-CoAs. So essentially, we have lost a third of the carbons that we originally had as a waste product, but we still have 2/3 of them left. We still have 4 in the form of 2 acetyl-CoAs that are unstable, and we need to continue to break this down. And it's going to continue on to the next step.
The citric acid cycle takes place in the matrix, or fluid, of the mitochondrion. This is where mitochondrial DNA is found and where fatty acid breakdown takes place. The citric acid cycle involves 8 chemical reactions that use acetyl-CoA and oxaloacetate to produce carbon dioxide, NADH, ATP, and FADH2. The NADH and FADH2 are electron carriers that can be used in the electron transport chain to make more ATP for a cell. Start of the electron transport chain. 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. One hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized.
Welcome to photosynthesis. Photosynthesis is the process by which plants use sunlight, carbon dioxide, and water to produce glucose and oxygen. The 2 main phases of photosynthesis are the light reaction and the Calvin cycle. The light reaction takes place in the thylakoid membrane, while the Calvin cycle, on the other hand, takes place in the stroma. The Calvin cycle is named after a scientist named Melvin Calvin. He was an American chemist who received the Nobel Peace Prize in 1961 for his discoveries. Water and light enter the thylakoid membrane and they produce oxygen as a waste product and also provide NADPH and ATP. In the stroma during the Calvin cycle, carbon dioxide goes in and glucose comes out. Let's talk about this in a little more detail. In the thylakoid membrane, there are 2 photosystems. A photosystem is a protein that has chlorophyll on its inside. Photosystem 1 and photosystem 2 are located in the thylakoid membrane. But before photosystem 1 takes place, photosystem 2 must initiate the reaction. Photosystem 2 is first, while photosystem 1 is second. But due to the fact that photosystem 1 was discovered first, it acquired the title of photosystem 1 over photosystem 2. The sunlight captured by these photosystems is used to power the movement of an electron through the electron transport chain until it reaches NADPH. Another way to think about this is by considering the electron transport chain to be a net, while the stroma and the lumen are the both sides of a table tennis table. The sunlight acts as the paddle that knocks the ball from one end of the table to the other, supplying it with energy and giving it its motion. While on the other side of the table is folded up, allowing the ball to bounce right back into its beginning state. That's an easier way to get an understanding of what goes on in the photosystems. In these photosystems, when they absorb H2O, they immediately break it down and split it. O2 is released, while hydrogen ions amass in the lumen. When we talked earlier and we spoke about light pushing electrons in through the electron transport chain, these negative electrons attract the positively charged hydrogen ions, and those force each other back out through the electron transport chain. But when the light hits that electron again, it comes on its own back through to pick up another one. And every time it goes back out of the electron transport chain, we're creating ATP. Next, we're going to talk about these products, NADPH and ATP. We're also going to talk about how they're used in the Calvin cycle as energy. The Calvin cycle takes place in the stroma of chloroplast, and it turns one carbon gas into a 6-carbon solid like glucose. In the Calvin cycle, there's a pre-assembled molecule containing 5 carbons called ribulose biphosphate, or RUBP for short. There's also rubisco, which is an initiator that welds one carbon atom from a carbon dioxide molecule with the RUBP chain to build a 6-carbon sequence, which then breaks up into 2 shorter chains with 3 carbons each. ATP provides the energy, while NADPH, adding 1 hydrogen to each of the 2 DGA chains created to make G3P. G3P is glycerolide triphosphate. Half of the G3P is used and the other half is reused to make RUBP and put back into the Calvin cycle. Without ATP or NADPH, this will not be able to work. That is the